Good afternoon, everyone, and welcome to the 10th webinar series of the Puerto Rico Outreach Program. We thank you all for being able to take the time to attend this presentation today. My name is Dr. Vivian Colon. I am professor at the UPR Graduate School of Public Health and co-leader of the UPR Outreach Program of the U54, UPR MD Anderson Partnership for Excellence in Cancer Research. Today, we have co-principal investigators and investigators of Full Project One, which will discuss with us insights, new findings, and future projects as part of this partnership. Thank you for Dr. Pettaway for being with us today. And with that, I'll leave it to you.
Hello, everyone. It's a real pleasure to be with you today. I'm Curtis Pettaway, Professor of Urology at the University of Texas, MD Anderson Cancer Center. And on behalf of my co-principal investigator, Dr. Magaly Martinez of the University of Puerto Rico, and our study co-investigators, Dr. Jad Chahoud, Dr. Xin Lu, and Dr. Niki Zacharias, we are really proud and honored to present the update of our research efforts on understanding and targeting convergent immunosuppressive pathways and molecular signaling in HPV positive and negative penile cancer. Next slide, please. I'm going to start today by basically giving you an overview of the therapeutic dilemma of penile cancer, and I'll talk a little bit about our study rationale and specific aims. And then we'll talk a little bit about our grant progress and novel insights. And you'll hear some specific talks by Dr. Xin Lu and Dr. Niki Millward on genetically engineered mouse models and human tumor xenografts that we're using to elucidate the molecular insights into penile cancer. We'll follow that with studies in fresh tissues and paraffin-embedded specimens from both Puerto Rico and the mainland US in talks by Dr. Magaly Martinez and Dr. Jad Chahoud. So we're using a variety of approaches to try to elucidate the biology of penile cancer to develop novel therapeutic insights. Next slide, please. So let's talk a little bit about the epidemiology. Penile cancer is a rare disease, especially in developed countries. It makes up less than 1% of cancers in the United States. And we estimate that approximately 2,100 new cases will be diagnosed, and there'll be 400 deaths annually. With respect to histology, squamous carcinoma is the most common type. So our research is mostly related to that. Now, in contrast, penile cancer is much more common in certain countries, especially those that are underdeveloped, where penile cancer can be seen in up to 10 to 20% of all malignancies in men. Just as an example, in Brazil, you see rates of penile cancer anywhere from 2.8 to 6.8 per 100,000, whereas in the US, that figure would be less than 1 per 100,000. It's a disease in elderly men, but it can be seen across the spectrum, as we'll show you. And the devastating thing is that this occurs in men who are sexually active. And in one series, approximately 30% of the men were less than 50 years of age. So it can be quite devastating. With respect to racial and ethnic predilection in the US, we see a similar incidence among Caucasians and African American men, but there is a disparity where Puerto Rican men of Hispanic ethnicity are disproportionately affected, with rates 2 to 3-fold higher than other non-Hispanic whites, other US Hispanic populations such as Mexican Americans and US Black populations. We see a similar thing with respect to the disparity with respect to mortality. So Puerto Rican men have a higher incidence and mortality from penile cancer. Next slide. Now, Magaly pulled some of the more recent data, I believe, from the Puerto Rican Cancer Registry here, and we see an increasing incidence with age. As you can see, the incidence goes up from 0.4 per 100,000 to those less than 50 to 14.3 per 100,000 to those men that are 80 years plus. Overall, again, you see a high incidence rate, 2.5 per 100,000, whereas in the other US populations, it was less than 1 per 100,000. And on average, it's estimated that about 50 men on the island of Puerto Rico will be diagnosed annually, and 8 men will die from the disease in any given year. Next slide. So when we look at what causes penile cancer, I think that the data coalesces around 2 things. One, exposure to things that promote inflammation, and then exposure to carcinogens. And just to give you an example, if you look at lack of neonatal circumcision, that's of course removing the foreskin of males when they're first born. When that's associated with poor hygiene, it leads to repeated bouts of inflammation and scarring, with then an inability to pull back the foreskin. So you get dirt and other kind of oils and things trapped under the foreskin, promoting chronic inflammation. This is associated with a 7 to 10-fold increased risk of developing penile cancer. Other inflammatory exposures include trauma, rash, a chronic inflammatory condition called lichen sclerosis. These men have penile cancer rates of anywhere from 6 to 8 per 100, or 6 to 8% when followed over 10 to 20 years. Now, with respect to exposure to carcinogens or carcinogenic agents, the number of sexual partners is directly related to the incidence of HPV infection. There are well over 100 different HPV genotypes. And I show here several of the more common that are associated with either viral warts or penile cancer. HPV in various meta-analyses have been shown overall to be associated with about 48 to 50% of penile cancer. And among the different genotypes of HPV, HPV 16 seems to be the most common one implicated in penile cancer, similar for cervical cancer, vulvar cancer, and head and neck cancer as well. Now, HPV is not only associated with carcinogenesis, it's also potentially a prognostic biomarker. We see on the graphs on the right-hand slide, the series above from the Netherlands, men followed over a long time. Those that were HPV positive had a better outcome than those that were HPV negative. And then in the series below from Germany, they measured P16, which we know is upregulated in HPV positive cancer, so it correlates with HPV infection. And what you can see is a better prognosis among those that were P16 positive. Now, we don't actually know why those men do better, but it may be related to sensitivity to various therapeutic strategies like radiation. Another carcinogenic product associated with penile cancer is tobacco products, whether it's cigarette smoke, chewing tobacco, or snuff. Can I have the next slide, please? So when we look at the natural history of penile cancer, it's shown first, you develop a primary lesion that just grows slowly over time. And the important thing here is that men tend to be embarrassed about having a lesion on the penis, and they don't often go to the doctor. So you often don't see these men till 6 months to almost 2 years sometimes before, after the lesion is growing. Now, what happens after this is that tumor emboli can break off from the primary tumor and travel to the regional inguinal lymphatics, like you see on that second picture down. And the disease can grow here. Now, importantly, if you control the disease in the primary site or in those inguinal region early on, these men can be cured of the disease. But when the disease becomes bulky, and you see in this last picture what happens in the uncontrolled growing. You get ulceration, you get massive infection, and this is often the cause of death from these patients, either sepsis or erosion of tumor into blood vessels. Now, while metastasis to distant sites like liver, lung, bone, brain can occur, they're not often the cause of death. It's usually regional complications. Can I have the next slide, please? So we looked at 10 series here, and you can see very clearly that those who are cured with surgery alone are those in that first bullet, those who have limited unilateral inguinal metastasis, generally of small volume, no extracapsular extension. They're cured of the disease with surgery alone. The next group, 3 or more unilateral, bilateral metastasis, some of these patients can be cured with surgery alone as well, but the cure fraction goes down. And then in the last 2 bullets, extranodal extension or pelvic lymph node metastasis, these patients are not cured with surgery alone, and clearly other therapeutic strategies combined with surgery are needed. Can I have the next slide, please? So this and if you could advance that next arrow for me, please. And the next one. Great, thank you. So we basically had studied the regimen paclitaxel, ifosfamide, and cisplatin in a prospective series before, and we saw that the response rate was about 50%. That means 50% of the men who got the drugs actually responded with downsizing of the tumor. So we followed that up with a longer term study in about 60 men, and we can see here that we treated largely advanced men, men with the N2 and N3 category of disease. And we saw responses in about 60 to 65% of these men, which was good. That means about 35 to 40% of the men don't respond. But in those responders, we were encouraged by the fact that responding to chemotherapy in association with surgery led to a durable complete response rate and survival at 5 years of about 50%, whereas with surgery alone, the survivals would have been drastically lower than that. Can I have the next slide, please? So the dilemma is that all patients don't respond. We just showed you that 35, 40% of the patients may not respond to the first line drug regimen, and 25 to 30% will even progress on the regimen. And unfortunately, surgery in that situation is of no benefit. Second, third line chemotherapy doesn't seem to benefit survival, as shown here on this graph, and chemoradiation is of no benefit either. So this is where we need novel therapy, and this is the focus of our grant. Next slide. So our overall objective here is to identify molecular patterns associated with HPV positive and negative penile cancer subtypes in the Puerto Rican and mainland US populations, and use this to potentially develop novel, promising therapeutic strategies for penile cancer based on these mechanistic insights gained from the clinical samples, genetically engineered mouse models, or our PDX human tissue models. We have preliminary data that in our genetically engineered mouse models that COX-2 is a master regulator of the inflamed tumor microenvironment. We've shown that a variety of immune cell populations are noted along with upregulation of PD-L1 in genetically engineered mouse models and in human samples. And based on expression analysis in Puerto Rican samples, we show novel upregulation of certain genes with HPV positive versus HPV negative samples. Can we have the next slide, please? So our central hypothesis is based on our preliminary data. And basically, the hypothesis that we're promoting is that chronic inflammation as a result of either HPV infection or downregulation of essential tumor suppressor genes, like Dr. Lu will show you, such as SMAD4 and APC, will promote the development of penile cancer. that key signaling hubs can drive penile cancer progression, including COX-2 and PDL1, and could be effective targets for immunotherapeutic intervention. And three, novel targets such as estrogen and Notch signaling that we've shown are upregulated in HPV positive cancer can play important roles in penile cancer progression. So then our specific aims are straightforward. We want to eradicate mouse penile cancer by combining targeted therapy and immunotherapy and potentially use that as a model of what we can do in humans. Two, identify the immunologic profiles associated with HPV infection in human penile cancer as potential therapeutic targets. And then three, potentially validate and target estrogen and Notch signaling in HPV positive penile cancer. Next slide. So we'll present then an update today on specific insights that we've made some progress on in our grant. So the first talks will be by Dr. Xin Lu, followed by Dr. Niki Millward on genetically engineered mouse models and human xenografts and how they're moving our research forward. And then we'll follow that with Dr. Magaly Martinez and Dr. Jad Chahoud. We'll talk about insights using fresh human tissues and paraffin embedded specimens, again, to accomplish grant objectives. Next slide. Okay, so now we'll hear from Dr. Xin Lu. Xin, go ahead and take it away.
Great. Thank you so much, Dr. Pettaway. So I'm Xin Lu. I'm a assistant professor at the University of Notre Dame. I'm an investigator in the U54 team. I'm really appreciative to that. So today I will discuss some results we recently obtained for the transgenic mouse model of penile cancer. Next, please. So there was no transgenic mouse model of penile cancer. So we developed the first one in the field. And this model is based on deletion of two important tumor suppressors, SMAD4 and APC, in the epithelial cell of the mouse penis. As you can see here, whereas the single knockout has either no phenotype or very mild hyperplasia phenotype, the combination deletion of the two tumor suppressors led to significant development of penile squamous cell carcinoma. And then on the right, you can see that we have these fluorescent lineage markers in the model. So the green nodules are the tumors, whereas the red colors represents the rest of the cells. And then you can also see that the tumors from the mice histologically really resemble the squamous cell carcinoma of the human penile cancer, shown here by the keratin pearls, for instance. Next, please. So with this model, we were able to identify some of the key molecular drivers for the disease. We performed the RNA sequencing to try to find the molecular patterns that are associated with the cancer development. As you can see here on the left, the heat map shows the dramatic contrast of gene expression between the wild-type mouse penis versus the SMAD4 APC or ICA tumors. But by network analysis, we identified two important drivers. One is the SOX2, which is a squamous cell carcinoma stem cell driver, stem cell gene. And then another one is a cyclooxygenase 2, COX2, which is a very well-known gene to drive inflammation. You can see both of the genes are highly expressed by immunohistochemistry study. Next, please. And then because SOX2 is a cell intrinsic driver for stemness, so we performed the ICGRNA-mediated knockdown of the gene in the ICA cell lines we derived from tumor models. And you can see the knockdown, we used the two independent knockdown, and then they both significantly inhibited tumor formation after we implanted the cells into the subcutaneous site of the mice. And this indicates that the cell intrinsic activation by SOX2 is an important component for the tumor development of penile cancer in mice. Next, please. And next, because the very strong COX2 activation in tumors, we speculate that the tumor probably have a very significant dysregulated inflammatory phenotype. So we did an immunoprofiling of the tumors compared to normal penile tissue using a CyTOF technology. And then you can see here, there's significant upregulation of the tumor infiltrating immune cells indicated by the CD45 staining. Next, if we separate the immune cells into different subsets, clearly there was actually overall downregulation of majority of the immune population, such as B cell, T cell, NK cell, dendritic cells. But there's one population that really shot up significantly. That's the CD11B and GR1 positive myeloid-derived suppressor cells, or MDSCs. And these cells are are immature myeloid cells that really drive immune suppression, as shown here on the right. You can see if we culture MDSCs with naive T cells stimulated with CD3 and CD28 antibodies, whereas the no MDSC co-culture, the T cell proliferate, indicated by the shifting of the peaks to the left. The co-culture with MDSC really stuck the peak on the right, indicating there's no proliferation of T cells. So those CD11B and GR1 positive MDSCs from the penile tumors are really suppressing myeloid cells. Next, please. In order to, this model really resembles the human penile cancer, not only at the histological level, but also by molecular signatures. So we did a co-analysis of the IC and ICP, which I didn't mention before, but this is the SMAD4 APC in addition to P10 knockout, triple knockout. The tumor is more aggressive and also resistant to cisplatin treatment. And then you can see that if we do co-analysis of the penile tumor for the mice with the penile tumor of the human, there is actually a very close resemblance of the transcriptome. And then also we can show that by the histological staining, the human tumors also highly express some immune-related markers such as CD3, CD68, and also PD-L1, as well as COX-2 and COX-2. So we think this GEM model is a very good, valid preclinical model for human PSCC. Next, please. So go back to the observation of the MDSCs. So because this model really resembles human disease, we really want to use this model as a preclinical platform to try some human-related clinical trials. So we performed the immune checkpoint blockade therapy with anti-PD-1 and anti-CTLA-4 combination. And then you can see that ICB treatment alone, the purple, is essentially similar to the blue color control. So there is no significant anti-tumor effect from ICB alone. However, if we combine this ICB with MDSC debilitating drugs, such as cabozantinib, which we previously published in the context of prostate cancer, or cocoxiclib, which is a known selective inhibitor of COX-2, in either case, you can see there is a significant combinatorial effect. And then the penile tumor of the mice are essentially either eliminated or significantly inhibited by this combination treatment. So I think this result is very provocative and really indicate that for immune checkpoint blockade to work in human advanced penile cancer, combination is necessary. Next, please. In summary, in this transgenic mouse model, the first one in the field, we show that there's a very strong cell intrinsic COX-2 mediated the tumor progression pathway, as well as extrinsic COX-2 mediated the inflammatory phenotype. And then we show that in this model, if we add a P10 knockout to activate the PS3K-AKT pathway, actually the tumor becomes resistant to cisplatin, which I didn't have time to show today. And then lastly, the anti-MDSC therapy can sensitize the tumors to immune checkpoint blockade therapy. Next, please. And this result was recently published by our team on Nature Communication. And then more importantly, I think this result is really clinically relevant because there is very recent publication on the combination of cabozantinib and nivolumab, which is a PD-1 antibody, in rare urological cancers, including 3 cases of penile cancer. You can see that in these 3 cases of penile cancer has passed many of the previous chemotherapies and become resistant. At least 1 patient become partial response and 2 become stable disease by this combination. And remember, this is also the combination that we used in our mouse models. Really reinforced the idea that these GEM models are very valuable to evaluate the efficacy of human clinical trials. Thank you very much. That's my presentation.
Hi, I'm Niki Zacharias Milbert, and I'm an assistant professor at MD Anderson Cancer Center, and I'm going to talk about the work we've been doing on patient-derived xenografts. A lot of this work was generated by our graduate student, Dr. Louis Segarra. He has an MD degree and he's in the process of getting his master's degree in biomedical sciences before he goes on to residency. Next slide, please. So patient-derived xenografts. In these models, we take patient tissue directly from cancer tissue directly from the patient, and then we engraft them into immunocompromised mice. And then after the tumor takes within the mice and the tumor grows, we go ahead and euthanize the mouse and then take the tumor and expand it into another set of immune-compromised mice. So what patient-derived xenografts allow you to do? Next slide. Let's say retain the heterogeneous nature of the tumor. This allows you to have a more clinically relevant model than, for instance, tumors generated using human cancer cell lines injected into a mouse. It also allows you to have more tissue for cancer biology studies, genomics, proteomics, or transcriptomics. Next, Magaly. These animal models can also be tested for different non-immune therapy regimens. So, currently there's, of course, Dr. Lu's transgenic models, but there are no PDX models currently in the literature. So there's definitely a need for more animal models of this very rare cancer. It also allows you to have more tissue to derive human cancer cell lines that you can use for in vitro assays. Next slide. Next. So in our case, we're generating animal models directly from resected penile cancer tumor tissue. So when Dr. Pettaway is in the OR and he removes the tumor is quickly taken to the lab where we process the tissue, and then we embed it into an NSG mouse right under the skin. And you can see this is what the, after the tumor engrafts, this is what the mouse looks like. And then at the time where we see the tumor has gotten to a certain size, we euthanize the animal, remove the tissue, and then we either use it for histology purposes, cryo-freeze it for molecular biology purposes, or we take it and embed it into another set of NSG mice. We currently have approximately 10 unique penile cancer animal models. We're in the process of comparing these models to patient tissue we've collected. And then, of course, we want to confirm that these animal models are identical to the patient's disease. Next slide. So currently, these are our current models that we have. As you can see, they're generated from a variety of different surgeries, from partial penectomies to radical penectomies to lymph node dissections. These models have different pathological staging. Many patients were treated with neoadjuvant therapy prior to resection, and then other ones of our models are therapy naive. We're in the process of showing that the genomic DNA through SDR fingerprinting is identical in the patient tissue compared to the PDX tissue. Next. So like I said, these are some of the first animal models of this rare cancer. We also have a mixture of P16 positive and P16 negative PDX models, so HPV positive and HPV negative. Like I said earlier, we have a mixture of therapy naive and therapy treated PDX models. And in fact, in one case from the same patient, we have therapy naive PDX model plus a therapy treated PDX model of the same patient. So we can look at the evolution that is occurring within the tumor tissue due to treatment. Next slide, please. So we'll continue, for our future directions, we'll continue doing the SGR genomic fingerprinting to confirm our PDX models are derived from patient tissue. We're in the process of also deriving penile cancer cell lines. And then we're doing immunohistochemistry staining of both the patient tissue and the PDX tissue to compare protein expression in both tissue types, specifically to look at estrogen receptor expression and also Notch expression. And then for our HPV positive PDX models, we'll look to determine the HPV genotyping of those particular PDX models. Next. And then we're going to test different targeted therapies in our HPV positive and our HPV negative PDX models to see if we can find a therapeutic benefit of these compounds. And now, next slide, please. And now I'll let Dr. Magaly Martinez Ferrer.
Well, thank you. And today I will be talking about the work that we have been doing in the identification of transcriptomic profiles in HPV positive and HPV negative penile cancer of Puerto Rican men. And the work that I will be presenting today has mostly been done by Maria Sanchez Vasquez, who is my research assistant. Recently, 2 undergraduate students joined the project as trainees. They are Desiree Arroyo Villegas and Yaris Roque Reyes, who are currently working in the project. Okay, so as Dr. Pettaway mentioned at the beginning, HPV is a risk factor for penile cancer, and the prevalence of penile cancer in Puerto Rico is significantly higher with the other populations within the United States. So the rationale of our study is that understanding the contribution and the molecular pathways by HPV to the pathogenesis of penile cancer is important for identifying new prognostic markers and therapeutic targets. So the objective of this project within the U54 Project 1 is to determine the genome-wide expression profiles in HPV-mediated and non-HPV-mediated penile cancer. So to conduct our study, we recruited patients. The patients that we recruited were diagnosed with penile cancer and underwent surgical intervention from 2015 to 2018 at the University of Puerto Rico ASEM Hospital. We had the clinical variables annotated for all the cases. We chose for this study 28 patients. Those samples were either paraffin embedded or stored at -80 degrees as fresh tissues. So after we recruited and collected the tissues, the first thing that we did was to do the HPV genotyping. We wanted to know what was the status of HPV on those samples. So we did DNA extraction. We did HPV genotyping using the InnoLipa kit that can detect 26 different types of HPV. We also did microarray analysis. We extracted RNA using fresh frozen tissues under the optimal conditions. And the microarray analysis was performed using Affymetrix GeneShip. From those 28 samples, there were 11 samples that were HPV positive and 17 samples that were HPV negative. After we conducted the microarray analysis, as I will be explaining, we identified genes that were differentially expressed, and then we wanted to validate those using paraffin embedded tissues from human penile cancers. And we did this using immunohistochemistry analysis. So this table here shows the clinical data associated with penile cancer cases. And one striking observation was the age of the patient. If you see at the top part of the table, the age at diagnosis, you can see that 18 patients from 28 were 50 years old or less. So there was a striking observation on how young the patients were. We also had the histologic subtype, the grade, stage, metastasis status, P16 status, and we will use all these clinical data to correlate with our findings. So this figure here shows the gene set enrichment analysis, or GSEA. As I mentioned, we use 11 HPV positive tissues and 17 HPV negative samples. And if you see at the left part, you can, those graph shows the enrichment status of the HPV samples related to estrogen response genes. So out of 50 hallmark gene sets, an interesting observation was that genes were enriched in the HPV positive samples for related to estrogen response genes. And these include the early response genes and the late response genes. If you see the graph, you can see that in the X axis, you see to the right, the HPV positive samples, and in the Y, you see the enrichment score, and you can see that they tend to go to the left, showing that they were more enriched there. And also another pathway out of 1,300 canonical pathways, the Notch pathway was significantly enriched in HPV samples also. So as I mentioned in the slide before, genes that are involved in the Notch pathway and estrogen pathway were differentially expressed between HPV positive and HPV negative cases. And in the graph here, you can see the normalized mRNA expression values for each of the genes that were differentially expressed. And why did we choose estrogen and Notch? First, because they were HPV positive samples were enriched on those pathways. And also estrogen contributes to the carcinogenesis and the tumor microenvironment reprogramming of cervical cancer, which is also a type of cancer that is HPV related. Also, estrogen receptors are expressed in penile endothelia and epithelia. From those genes, we found that nuclear receptor coactivator 3, or NCOA3, and RAF1 proto-oncogene, or CRAF, were enriched in HPV positive samples. And on the other hand, when we look at Notch, studies have shown that Notch activity can promote prostate cancer cell proliferation and survival, cell migration and invasion, and aggressiveness. However, there's also suggested that it might have a tumor suppressive role. So for Notch, we found that JAK1, Notch2, and ADAM17 were all overexpressed in HPV positive samples when compared to HPV negative samples. So after we found this overexpression, we wanted to confirm those results, and we used immunohistochemistry analysis of paraffin-embedded human penile cancer tissues to stain them with the antibodies and score their expression. So here you can see that in the panels, the immunohistochemistry staining for HPV negative tissues and a representation for HPV positive tissue. And in the right side, you can see a graph that shows the expression of HPV of Notch2 in HPV positive samples were significantly higher when compared with the HPV negative samples. So we were able to validate the overexpression of Notch2. The same way happened with JAK1. You can see again here the pictures of the immunohistochemistry analysis for HPV negative and positive. And in the graph, you can see also that the HPV positive samples showed overexpression of JAK1 when compared with the HPV negative samples. However, when we did immunohistochemistry analysis for ADAM17, as you can see here, for HPV positive and HPV negative, and for RAF1, they were not differentially expressed when we look at HPV positive versus HPV negative tissues. So in conclusion, from the data that we have until now, we found that our transcriptomic data reveal extensive signaling changes associated with HPV in penile cancer among Puerto Rican men. The overexpression of Notch2 and JAK1 related to Notch signaling was confirmed in HPV positive penile cancer using immunohistochemistry analysis. And our results can help in the development of diagnostic and prognostic biomarkers for HPV positive and HPV negative penile cancer. So some things that we're currently doing and some future directions are we will perform immunohistochemistry analysis of human samples that we will receive from MD Anderson Cancer Center and validate the results that we got from the samples in Puerto Rico. We will also validate results using Western blot analysis. We will perform functional validation of the estrogen signaling and NOx signaling in HPV positive penile cancer. We will do correlation of the positive expression of genes and proteins with HPV status and the clinical variables, and will identify the immunologic profiles associated with HPV infection in human penile cancer. So now Dr. Chahoud will follow on.
Thank you, Dr. Martinez. Thank you for the opportunity to speak to you today. I'm Jad Chahoud. I'm an assistant professor at the GU department and Moffitt Cancer Center, and with a focus on penile cancer research. I'll be taking you over some of our preliminary results from looking at the tumor immune microenvironment on some of the human samples that Dr. Pettaway had prospectively collected at MD Anderson. Next slide, please. So as Dr. Pettaway mentioned, our current understanding and current knowledge of the immune environment of penile cancer is kind of limited. We know that PD-L1 expression tend to be positive by simple immunohistochemistry in around 40 to 60% of reported studies in penile cancer. Even though PD-L1 expression has been a potential biomarker in some other solid tumors like lung cancer and head and neck cancer, it has not been the case in other cancers, and PD-L1 expression is part of the whole picture. As we know, the immune contexture is defined by the type, functional orientation, density, and location of the different adaptive immune cells within each distinct tumor region. These vary significantly between tumor types, and the immune contexture in penile cancer has not been yet thoroughly investigated. In the literature, in the general solid literature, there has been 3 lymphocyte infiltration forms that have been distinguished. First is the immune inflamed phenotype, which is characterized by tumor infiltrating lymphocyte high density. The immune excluded phenotype, which shows the lymphocyte located mainly at tumor stroma interface, and an immune desert phenotype that lacks lymphocytic infiltration. Furthermore, there is a plethora of other immune cell subpopulations that have been shown recently to have a prognostic markers for immune therapies with different myeloid cells like macrophages, type 1 and type 2, and different myeloid cells. Next slide. So for this reason, we aim to conduct this study that was conducted at a single center using prospectively collected tissues that had enough tissue to conduct multiplex immunofluorescence using 5 immune markers: CD3, CD8, CD68, PD1, and PD-L1. So the benefit of this type of multiplex immunofluorescence is at 3 levels. First, we don't require a significant amount of tissue. 2, it should be less time consuming, as you can test for different markers at the same time. And 3, most importantly, it allows us to understand the different interaction between these markers at the different tumor location. Also in our study, we had HPV testing by P16 immunohistochemistry and HPV PCR COBAS. Next slide. So here we highlight the algorithm for description of the composite imaging available to our pathology team when defining the density of each of these subsets at both the tumor and stromal region. And as you can see, we have the description of the different T cell subset with the subset with their PD-1 and PD-L1 expression, and also we have a description of the macrophages with CD68 and their PD-L1 expression. Next slide. So as I mentioned before, we evaluated a total of 54 tumor tissue from the prospectively collected cohort at MD Anderson that had enough tissue to allow us for testing. And as you can see, the patient characteristic, our cohort was significantly enriched in Hispanic with around 41% of the population. We had 70% of the patient population be HPV negative, which is reflective of what we think is the numbers in the US. And also the cohort was evenly distributed between lymph node negative and lymph node positive on pathology. Next slide, please. Here we highlight these images show some of the multi-immunofluorescence composite imaging. And I wanted to bring those two examples. So because they appear to have on our prelim analysis some significant value, prognostic value. First, comparing a case of high tumor intensity and density of CD68 positive cells that are highlighted in yellow in figure A versus figure B that has a low tumor macrophage density where you cannot see any yellow. And the second sets of figures with figure C first showing a high, basically high stroma infiltrate of exhausted T cells, which are cytotoxic T cells, CD3, CD8 positive cells that express PD-L1, in comparison to basically high in cytotoxic T cell stromal tumor margin, where you don't see that bright orange PD-L1 expression on the cytotoxic T cells. Next slide, please. The reason why I brought them up, because it appears when we conducted multivariate analysis that cases that had high CD68 density has significantly improved disease-free survival and overall survival and cancer-specific survival. It's important to mention that our panel was not able to differentiate between M1 and M2 macrophage types because we did not include markers that allows us to conduct that differentiation. That could be an important thing to be performed in the future. Next slide. Also, cases that appear to have a low stromal expression of cytotoxic exhausted T cells has significantly better disease-free survival and overall survival, and the numerical trend to improved cancer-specific survival. Next slide. So in conclusion, this is the first study using the novel multiplexed image analysis to assess the immune microenvironment in primary penile cancer. We show that elevated macrophage density in the total tumor was associated with significant lower risk of recurrence and improved overall and cancer-specific survival. And also, it appears to be that the presence of exhausted cytotoxic T cell at the tumor stroma region appeared to be associated with worse survival. We did not differentiate. We did not find any significant difference between the HPV negative and HPV population when it comes to the immune microenvironment description and prognostic value of these markers, which probably would allow us to conduct in the future all-inclusive immune-targeted clinical trials with the therapeutic goals. Next slide, please. So as I alluded to in the future, we aim to look at the immune spatial analysis, because this is one of the values of conducting multiplex immunofluorescence analysis. We are trying to optimize a panel 2 with an M1, M2 that allows to differentiate M1 from M2. Also, we will have multi-omic data available from human tissue that we could further analyze and drive even more some of those hypotheses. And we hope to validate these findings in prospective larger cohorts. And I leave it to Dr. Martinez for the acknowledgement.
Okay, thank you. Yes, so this project is possible thanks to the collaboration and collaborative group that we have between co-leaders and co-investigators. And each group has its own collaborators that make possible the progress of this project. First, the PDX team that is composed of Dr. Pettaway, Dr. Zacharias, Dr. Lu, Dr. Segarra and Rao, and their support staff, Sue Martinez, Tapati Maiti, and Angelina Alanis. In the USA, the human studies are possible with the collaborators, Dr. Gabler Neto, Lascano Segura, Roger Parra, Rao, and Wistuba. Also, funding of Dr. Jad Chahoud from the 2018 Conquer Cancer Foundation Young Investigator Award, from the MD Anderson HPV-related cancers Moonshot Program, the Cancer Genomics Laboratory, and the Comprehensive Partnerships to Advance Cancer Health Equity, U54 from the NCI, Center to Reduce Health Disparities, to Dr. Pettaway. And here in Puerto Rico, our collaborators, Dr. Antonio Puras, who is the urologist, clinical urologist here that made possible all the collection of the human samples. Dr. Maria Marcos, he is the pathologist. Dr. Josue Perez, Ortiz, and Nogueras that helps with the microarray and statistic analysis. Both in the US and Puerto Rico, we thank profoundly the patients that agreed to give us tissue samples in order to conduct our studies, and at the University of Puerto Rico Comprehensive Cancer Center for their support. And here we are the co-principal investigators and co-investigators. If you have any question related to the content of today's seminar, you can either email Dr. Pettaway. The email address is under Dr. Pettaway picture. You can email me, and we will either answer or direct any questions to any of the co-investigators that talked today. So thank you very much for your attention.